Good morning, everyone. Welcome to today's presentation. I'm Dr. S.K. Singh, and I'm excited to talk to you about biological oxygen demand, or BOD. In this session, we'll explore what BOD is, why it's important, and how it's measured. Over the next few slides, I'll guide you through the key concepts and applications of this vital environmental parameter. Let's get started. Let's talk about biochemical oxygen demand, or BOD for short. Essentially, BOD measures how much dissolved oxygen in water is consumed by aerobic microorganisms as they break down organic matter. This process happens over a specific period and at a set temperature, making BOD a helpful indicator of organic pollution. When we express BOD in milligrams of oxygen per liter, or mgO2L, we're quantifying how much oxygen is used up during decomposition. Now, why is this important? Well, BOD serves as a key indicator of the level of organic pollution in both water and wastewater. High BOD levels suggest there's a lot of organic material that microbes are working hard to decompose, which can lead to oxygen depletion in the water. This makes BOD a critical parameter in environmental standards and discharge permits, helping regulators ensure that water bodies are protected from excessive pollution. So, understanding BOD helps us monitor water quality and enforce environmental protections. It's a vital piece of the overall water management puzzle. Let's take a closer look at the standard test, norms commonly accepted in water quality assessment. One of the most widely used methods is the BOD-5 test, which measures the 5-day biological oxygen demand at 20 degrees Celsius. This method is the international standard for regulatory monitoring, because it provides a consistent and reliable way to assess organic pollution in water. When we talk about standards and references, one key document is the standard methods for the examination of water and wastewater, specifically Method 5210B. This method is broadly accepted by environmental regulators around the world, making it a cornerstone in water quality testing. In addition, ISO and N standards also use similar conditions for their testing methods, which helps promote harmonization and comparability across different regions and laboratories. Now, from a regulatory standpoint, the choice of a 5-day incubation at 20 degrees Celsius is quite deliberate. It strikes a good balance between practicality, since longer tests can be cumbersome, and biological activity, since this temperature and duration effectively reflect the microbial response to organic matter in water samples. So, in essence, these norms ensure that our testing is both standardized and meaningful, allowing us to reliably monitor and compare water quality across different settings and over time. So, why do we measure BOD, or biological oxygen demand? Well, the main reason is that it helps us understand the amount of biodegradable organic material in water. This is really important because it directly impacts the dissolved oxygen levels, which are vital for aquatic life. If there's too much organic matter, it can lead to oxygen depletion, harming ecosystems. Measuring BOD also allows us to evaluate how effective wastewater treatment plants are. If treatment is working well, we should see lower BOD levels in the effluent before it's released into the environment. And speaking of the environment, measuring BOD is crucial for protecting receiving waters. By keeping track of BOD levels, we can prevent oxygen depletion damage downstream, ensuring healthier water bodies. Now, just to give you a sense of typical BOD ranges, unpolluted natural waters usually have BOD values around 0.5 to 6 milligrams per liter. Treated effluent often falls between 10 and 30 milligrams per liter, indicating some organic material remains, but is generally manageable. Raw sewage, on the other hand, can have BOD levels from 150 to 400 milligrams per liter, reflecting its high organic content. So, by monitoring these levels, we can better manage water quality and protect aquatic ecosystems effectively. Let's take a closer look at the BOD test principle. The process starts with taking a sample, which is often diluted to ensure accurate results. This sample is then incubated with microorganisms, which are essential for the test. During incubation, these microorganisms respire, meaning they consume dissolved oxygen, or dew, as they break down organic matter in the sample. The key idea here is that the amount of oxygen they use up reflects the amount of biodegradable organic material present. Over the 5-day incubation period, we measure how much dew decreases. That drop in oxygen is directly related to the BOD, or biological oxygen demand, of the sample. Now, to get consistent and reliable results, we follow standard conditions. The temperature is maintained at 20 degrees Celsius, plus or minus 1 degree, because temperature affects microbial activity. The incubation lasts exactly 5 days, giving enough time for the microorganisms to do their work. And importantly, the process is carried out in darkness. This prevents photosynthesis, which could otherwise produce oxygen and skew our results. So, in essence, the BOD test is a practical way to gauge how much organic pollution is in water, by observing how much oxygen-consuming activity occurs over a set period under controlled conditions. All right, let's go over the apparatus and materials you'll need for this experiment. First, we have the 300 ml BOD bottles with ground glass stoppers. These are specially designed to minimize oxygen entry, which is crucial for accurate BOD measurements. Next, you'll need a dew measurement setup, which can be either a Winkler titration kit or a dew meter, depending on what's available in your lab. Then, we require an incubator set to 20 degrees Celsius to maintain a consistent temperature during the incubation process. It's also important to have dilution water that's saturated with oxygen, so your measurements remain accurate. Lastly, if your experiment involves adding nutrients to seed samples, you'll need nutrient solutions on hand. All these items are essential to ensure your experiment runs smoothly and yields reliable results. They form the foundation for accurate and consistent BOD testing, which ties back to our overall goal of assessing water quality effectively. When collecting and preparing samples, it's crucial to ensure they truly represent the water source we're testing. That means taking a sample that accurately reflects the conditions of the entire system. Timing is also important. Analysis should ideally start within 2 hours of collection to prevent any changes in the sample. If that's not possible, refrigerate the sample at around 4 degrees Celsius to preserve its integrity. Another key step is removing residual chlorine from the sample, a process called dechlorination. Chlorine can kill the microbes we're trying to detect, so if it's present, it can lead to inaccurate results. Lastly, adjusting the pH to roughly 6.5 to 7.5 before incubation helps create optimal conditions for microbial growth, which is critical for reliable testing outcomes. All of these considerations are part of good sample collection and preparation practices, which underpin the accuracy of our testing and help us make sound decisions about water safety. Let's talk about dilution and seed considerations. First, when should we dilute our samples? Well, if we expect the BOD to be high, it's a good idea to dilute the sample. This helps ensure that the dissolved oxygen doesn't get completely consumed during incubation, giving us more accurate results. Now, moving on to seeding. Sometimes, the sample might not have enough microorganisms to produce a reliable BOD measurement. In those cases, we can add a known seed, like standard wastewater, to boost the microbial population. It's also important to run seed control bottles alongside our samples. These controls help us account for the oxygen consumption caused solely by the seed itself, allowing us to correct our measurements accordingly. Overall, these steps, dilution and seeding, are essential to get precise and meaningful BOD results, fitting into the bigger picture of accurately assessing wastewater quality. Let's talk about measuring dissolved oxygen, or dew, in our samples. First, it's important to measure the initial dew, or DOI, right after you've prepared your sample. This gives us a baseline of how much oxygen is present at the start. Then, after keeping the sample at 20 degrees Celsius for 5 days, we measure the dew again, which is our final dew, or DOV. This helps us understand how oxygen levels change over time, which is crucial for assessing water quality and microbial activity. When it comes to actually measuring dew, there are a couple of reliable methods. One is the Winkler iodometric titration, a classic and highly accurate chemical method. The other is using a dew probe, or electrode, which needs to be properly calibrated to ensure precise readings. Both methods are effective, but choosing the right one depends on your specific needs and equipment availability. Connecting this to our overall theme, these measurements of dew provide important insights into the health and stability of aquatic environments. Monitoring changes from initial to final dew helps us evaluate biological activity and oxygen consumption, which are key indicators of ecosystem health. Let's take a closer look at BOD5 calculation and its basic formula. This is a fundamental concept in wastewater treatment, as it helps us determine the amount of oxygen required to biologically break down organic matter. Understanding this formula is key to designing effective treatment processes. Essentially, the basic formula allows us to quantify the BOD5, or the biochemical oxygen demand over 5 days, based on the amount of organic material present. It's important because it directly relates to how much oxygen we need to sustain microbial activity. When we connect this to the overall theme, it reminds us of the importance of accurate measurements and calculations in managing water quality efficiently. So, grasping this basic formula is a stepping stone to mastering more complex treatment processes. Now, let's talk about the use of dilution factors. When we perform BOD tests, we often dilute our samples with water to get accurate readings. The key here is understanding how to calculate the dilution factor, which we denote as P if, for example, we take a certain volume of our sample, say X milliliters, and add it to a total volume of 300 milliliters in the bottle, then our dilution factor P is simply X divided by 300. To make this clearer, imagine we have 15 milliliters of sample in that 300 milliliter bottle. The calculation for P would then be 15 divided by 300, which equals 0.05. This number tells us how diluted our sample is compared to the total volume. Once we have this dilution factor, we can correct the BOD 5-day value, BOD 5, by dividing the difference between the initial dissolved oxygen, DOI, and the final dissolved oxygen, DOV, by P. Let's look at an example. Suppose our initial dew, DOI, is 9.2 mg per liter, and our final dew, DOV, is 2.1 mg per liter. If no seed was used and no further dilutions were made, then our calculation for BOD 5 is straightforward. Subtract DOV from DOI, which gives us 9.2 minus 2.1, resulting in 7.1 mg per liter. This value, around 7.1 mg/L, suggests a moderate level of organic pollution in the sample. So the key takeaway here is that understanding and applying the dilution factor correctly is crucial for accurately interpreting BOD data, especially when samples are diluted to ensure precise measurements. When it comes to quality criteria and ensuring valid results, regulatory labs have specific acceptance standards that we need to meet. First, the final dissolved oxygen, or DOO, should be at least 1 milligram per liter. This ensures that the oxygen levels were sufficient during testing. Additionally, the difference between the initial DOO, or DOI, and the final DOO, DOV, should be at least 2 milligrams per liter. This change indicates that microbial activity has taken place, which is essential for accurate measurements. Temperature control is also critical. Keeping it consistent at around 20 degrees Celsius, within a plus or minus 1 degree margin, helps ensure reliable results. Now, we also need to be aware of potential interferences. For example, toxic substances like chlorine or heavy metals can inhibit the microbes, skewing our results. And exposure to light or the presence of algae can also affect the readings, especially if bottles are not kept in the dark. All these factors are important because they help us produce accurate, reliable data, which ultimately supports the integrity of our entire testing process and aligns with regulatory expectations. Now, let's take a look at some advanced or alternative methods for measuring BOD. One of these is the use of respirometric or BOD analyzers. These devices provide automated, direct readings based on oxygen uptake, which makes the process faster and often more accurate. It's a significant step up from traditional bottle tests, especially when quick results are needed. Another important method is the carbonaceous BOD, or CBOD. This technique uses nitrification inhibitors to isolate and measure only the carbonaceous demand, which is particularly useful in situations where nitrogenous demand is regulated separately. Essentially, it helps us focus specifically on the organic carbon-oxygen demand, without interference from nitrogen-related processes. These methods are often accepted within regulatory frameworks, especially when rapid results or specific data are required. They provide a practical and efficient way to meet regulatory and treatment process needs, aligning well with the overall goal of optimizing wastewater management. Let's wrap up with some key takeaways. First, BOD5 remains the standard method for assessing the organic load in both water and wastewater. It's a reliable way to gauge how much biodegradable material is present. Next, it's crucial to remember that the test conditions, such as temperature, incubation time, and dissolved oxygen measurement, must be strictly controlled. Any deviation can lead to inaccurate results, which could impact your treatment decisions. Finally, these results are essential for ensuring compliance with environmental discharge norms and for evaluating how well your treatment processes are performing. Keeping these points in mind helps us better understand and manage water quality effectively. For further support, please don't hesitate to reach out to us. We're here to help and provide any additional information you might need. Feel free to contact us through the channels provided, and we'll be happy to assist you. Thank you very much for your time and attention today. We appreciate your engagement and look forward to supporting you moving forward.